Hi, my name is Henry Jose. This is my lab gut microbiome project. The microbiome is the group of microorganisms that inhabit the environment. The gut microbiome is specifically the bacteria that inhabit the gut region of our bodies. They have been proven to be linked to physiological conditions. For example, a study conducted researching on 1,054 humans in the Flemish Gut Flora project. It found that the gut bacteria produce compounds that act on the nervous system. One thing found was that the coccyx and dialister were linked to mental illness, being more common in people without depression because of their ability to produce DOPAC. This was correlative relationship found in the study, along with other types of bacteria that were found to be negatively or positively correlated with mental health. Another study was conducted by a neurologist on Parkinson's disease patients, and it found that an abundance of Parvitellus in feces of PD patients was reduced by 77.6 as compared with controls. The relative abundance of enterobacteriaceae was positively correlated with postural instability and gait difficulty, which are results of Parkinson's disease. We wanted to conduct our study pertaining to gut microbiomes. Our question was will similar weight have an effect on the gut microbiome? And our hypothesis was that the gut microbiome. Pertaining to the project, I will be giving a brief overview of the two species we compared. In our study, two examples of huskies, Maya and Nori, and two examples of pugs, Buster and Cooper, were compared. All four are types of dogs. The huskies are both female and the pugs are both males. Both of the pugs are 2 years and 2 months old. Maya is 3 months old, while Nori is 2 years and 5 months. Nori and Buster both weigh 37 pounds, while Maya weighs 19.1 pounds, and Cooper weighs 35 pounds. The only dog with health issues is Maya. She has coccidia. She also the only dog that stays indoors. The other dogs go indoors and outdoors. Both of the huskies exercise a lot, and both pugs exercise occasionally. Cooper and Buster have both eaten Neutral Ultra for their entire lives. The huskies' diets vary from each other. Nori has had a raw diet for 1.5 years, and Maya has had grain-free dry for 2 months. For this section, I'll be giving a brief overview of what our materials and methods we used for our project were. Once we had chosen and identified our samples, we had to purify the DNA from the fecal matter by using the Qui-Gon kit from Qui-Gon Inc. And following the manufacturer's instructions, by following the instructions, we washed away impurities from the stool sample, leaving us with the isolated DNA. We then made DNA that we isolated 10 times more concentrated by using the Zymo kit from Zymo Inc. Follow the manufacturer's instructions by eluted our isolated DNA and DNA elution buffer at a volume that was 1/10 of our initial volume. Once we had concentrated our DNA, we wanted to quantify the isolated DNA and estimate any contamination that in proteins and RNA, measuring its purity. We found out using the nanodrop spectrophotometer. We measured the UV light that the DNA absorbed from the nanodrop spectrophotometer at 260 nm and used the ratio of absorbance at 260 nm/280 nm to measure the purity of our DNA. We recorded the DNA concentration 260/280 ratio. When making sure we had a high enough concentration of DNA extracted from SCAD, we wanted to make sure that the concentration of DNA we had was purely DNA and not contaminated with proteins, because this would interfere with our results. We knew all of our concentrations were high enough because we compared our 260-280 sample ratios to acceptable pure DNA ratio range. Our ratios of 1.80, 1.85, 2.07, and 1.50 were all within the range. Sample 310 had an initial DNA quantification of 92.8 ng and a 260 to 280 ratio of 1.80. This ratio is the pure DNA range of 1.5 to 2.2. Sample 518 had an initial DNA quantification of 21.2 ng and a 260 to 280 ratio of 1.85. This ratio is in pure DNA range of 1.5 to 2.2. Sample 593 had an initial DNA quantification of 92.7 ng and a 260 to 280 ratio of 2.07. This ratio is this ratio is in pure DNA range of 1.5 to 2.2. Sample 863 had an initial DNA quantification of 1.9 ng and 260 to 280 ratio of 1.50. This ratio is in the pure DNA range of 1.5 to 2.2. We use agarose electrophoresis to check the products out of PCR reactions and determine the size of the DNA fragments that we were analyzing. The smaller DNA fragments migrate further, the longer ones during the procedure, because the distance it migrates inversely proportional to its molecular weight.
I think it's next.
For the negative control, you can see there was no band present. For the positive control, there's a band present. For sample 310, there's a band present. For sample 518, there's a band present. There is no band present for 593, and for sample 863, there is no band present. We expected the size of the amplicon to be approximately 550 bb. The presence of a band 550 bb for the positive control lets us know that DNA amplification worked. The absence of a band for the negative control is also a good sign, because negative side should not contain any DNA. The presence of a band for samples 310 and 518 shows that the amplification worked, but since there is no band for samples 593 and 863, this shows that the DNA amplification did not work. We use the Agent Core Ampere Kit, which is a PCR cleanup kit. We followed the manufacturer's instructions in order to clean up the amplicon for our amplification PCR reaction. This kit cleaned up by removing components of the amplification PCR buffer, which included Mg2 plus DNTPS and enzymes. It also removed the forward and reverse primers that weren't used. We wanted to remove these molecules because if they remained with the amplicon, it would interfere with the indexing PCR by binding to the indexing PCR primers. To perform the indexing PCR reaction, we placed our PCR tubes in the thermocycler. First, it was set to 95 degrees Celsius for 3 minutes. Then it went to 8 cycles for 95 degrees Celsius for 30 seconds, 55 degrees Celsius for 30 seconds, and 72 degrees Celsius for 30 seconds. After these cycles, we set it at 72 degrees Celsius for 5 minutes. Using an indexing PCR reaction, we were able to add a unique set of sequence tags to the end of the amplicon from each sample, which was done to help us identify our samples when we sequenced DNA. After indexing our samples, we performed a second PCR cleanup, this time using the Agent Core Ampere XP kit to clean up the amplicon from our indexing PCR reaction. This time, the cleanup removed the single-stranded DNA of the primer's DNTPS buffer and dye so that it would not interfere with the DNA sequencing analysis. Once the amplicon was cleaned up again, we used the fluorometric assay to determine the concentration of our amplified sample that we were left with after the cleanup. We used the fluorescent dyes and measured the fluorescein using a Quebec fluorometer. For our fluorometric quantitation, our molar concentration for sample 310 is 95.3, sample 518 5.95, and for our positive control 16.4. We can see that we lost sample 593 and 863 during the quantitation process. We use a fluorometric assay instead of the UV spectrophotometer assay that we used earlier, because the fluorometric assay provides a more sensitive, selective, precise measurement, which helped when we prepared the dilutions for the sequencing reaction. We took our fluorometric reading, which were measured in nanograms, and divide this number by 0.363 to get our DNA nanometers concentrations. As long as the concentrations can give us a final 100 for a nanometer solution, we were able to make our dilutions for DNA sequencing. Dilution of our amplified DNA was prepared. We were ready for DNA sequencing. The nucleotide sequence of each indexing PCR fragment was determined using an Illumina MSEC next generation DNA analyzer. Here are successful reads we got. So for our quality filter, we had a successful read of 8,336 for our positive control, 8 for negative control, 6,315 for our sample 671, and for our sample 672, we had 7,452. They all passed the quality filter. We had to pick new animal samples due to samples 310 and 518 not having analyses available, so we now have samples 671 and 672. The positive control had 8,000 successful reads, and of these reads, 99.99%, 8,000 of them were a type of bacteria at the species level. The most abundant bacteria found was Escherichia alberti. So right here we have the kingdom level, the order level, and the genus level. At the kingdom level, all samples contain bacteria. This is expected, but as the taxonomic level became more specific, we started to see differences. At the order level, the positive control consists of enterobacteria, but samples 671 and 672 consist of Bacteroideas. At the genus level, the positive control consists of Escherichia, while samples 671 and 672 consist of Prevotella. Prevotella has been found to cause periodontitis, which is inflammation of the gums. This data was collected to answer my question of whether similar weight would have an effect on the gut microbiome. Our hypothesis was the gut microbiome would be the same, and our data does support this.